Okay, I'm just going to go through a few exemplars of some cell diagrams. You might recognize some of your own ones in here. Now, just a warning there, I'm not trying to criticize your drawings, and I'm not trying to, I've made sure there aren't any names on them, but I'm just trying to make sure you don't actually lose any marks when you do these things. And actually, it's usually those students who do the most practice and have the most mistakes or identify the most mistakes along the way who then don't make the mistakes in the actual assessment. So making these mistakes along the way is actually often a good thing, and if you're aware of them, then that's going to help you get a better mark. So it's often those students who do the most practices or go through it the most who, yes, there's lots of little things that could be improvements, but they tend to get better marks in the end. So I've tried to take some of these exemplars because they're really good examples for other students too, to hopefully prevent them from making those mistakes. So please don't feel I'm trying to criticize your work. No one else should really be able to figure out or know that it's yours anyway. So anyway, let's get into it. So here off, my first example, some onion cells. Few things. You'll notice the nuclei are shaded, so that's a big no-no. You could stipple, but really they should just be a circle. We've got some quite sharp corners here, and I don't know if that's really representative of their true shape. Here the pencil is probably not quite sharp enough, it's a little bit feathery. So by feathery we mean you can see little white dots or areas within the line. It's not one black line, and here the junction where they've connected up the two lines here isn't particularly great either. The proportion is pretty good. They've used the space, so that's pretty decent. The general shape of the cells is okay, so not too bad. Missing labels. I think when we were doing these in class, the focus was on the actual drawing, so I wasn't too strict on that. And the title should be underlined. Alright, looking at this next one, so again, maybe pencil isn't just quite sharp enough, although that could be when I scanned in these images. Sometimes the scanning does that a little bit too. One thing here, I think the labels, that's good, could have labeled the cytoplasm. The shape of the cells here is a little bit sort of misleading. The cells really are, do have sort of a roughly six-sided shape when we're looking at onion cells, and that should be reflected in the picture. Otherwise, this is a pretty good diagram. The other thing is this cell here has kind of been latched on. You can see it's just been added on by going like this. All right, it doesn't. This line probably should have followed right around, and it probably wouldn't have had this little spiky bit here, but it may have. So it's not too bad. Alright, this one. Okay, so definitely a couple of issues here. The shape of the cells is good. Probably should have drawn in a fourth cell, but that's okay. The cell wall here. Now it's almost like this one has been drawn with straight lines. And although onion cells often have that roughly six-sided shape, they're probably not really as straight lines as they're shown here. The other issue is a lot of these lines don't connect up, or in other places they sort of touch. Also, this cell, for instance, at the top here loses its cell wall entirely where it meets the other cells. So you really need to draw each cell individually in its full detail. If you can't see any nuclei, try adjusting the focus slightly. As you adjust the focus, you're essentially focusing at a different depth. So you're kind of moving up and down through the specimen as you focus. And you'll probably find you can still see nuclei somewhere even if it's at a slightly different depth. You wouldn't have to draw a nuclei in every single cell if you can't see one, but you should be able to see a nucleus in at least one of your cells, at least with onion cells. If you can't see them in any of your cells, at the end of the day, draw what you can actually see. So here again, maybe also not quite the sharpest of pencils, but needs to be much more careful with the junctions. And also, like I said, each cell should have its own cell wall. If the cell wall isn't very thick, it also needs to be in proportion to what you see. You might just want to draw it as a single line. If it is thick enough that you can draw it, and that's going to depend on how big you're drawing the cells, and basically how thick the cell wall is. It depends on the cell types you're looking at. So if it is significantly thick, you can draw it in, but I'm guessing in this case the cell wall wasn't this thick in proportion to the overall cell. So that's a little bit out of proportion too. Reasonable size, that's pretty good. Title magnification, that's great. Could have had a few more layers, a few more labels like nucleus. I guess they didn't draw one, so you can't label it. And cytoplasm could have been labeled too. All right, here. So here the pencil seems reasonably sharp, which is good. There's almost looks like there's a tiny little gap here. Probably get away with that. The main issue here is the students tried to avoid having to deal with those junctions where the cells meet each other in the corners and things by just doing one on top of the other. And unfortunately you can't get away with that. So you need to actually do them in a group. So like I said in class, I recommend that each cell is touching at least two other cells. I mean, obviously, if you're drawing like blood cells or something and they're floating free and they're not actually touching at all, then again you draw what you actually see. But if it is like onion cells, if they're arranged, your diagram should reflect or show that arrangement. So it's not just showing the details within a cell. That's why you draw more than one cell. It's showing how they're sort of arranged or fit together too. Labels is good. Probably should have done those in pencils too. Title should be underlined, magnification included, and again the shape of the cells is a little bit they're not quite so rounded, and here we've got a little bit of that latching on again. Okay, here the walls are a little bit rough. So plant cells or onion cells tend to have a bit more of a smoother cell wall, although I've seen students do the opposite. When we do cheek cells, they've been scraped off, so they're often a little bit damaged, and they have a very sort of jagged or rough edge to them, and students often draw them as nice round smooth circles. Now if it's got sort of a rough edge, you don't need to follow the exact contour of that rough edge, so you don't need to show every little zig or zag or crevice. But you might want to give it a bit of a random sort of roughness, because you're just trying to illustrate what that cell actually looks like, or what a cell of that type looks like. So here it's kind of the other way around though. It probably should have been a bit smoother. This junction here you can see is a classic example of this lines. This cell's been sort of latched onto the other one, and this line's come around and it's gone over. So it just sticks out the other side a little bit, and here two lines that don't quite connect. Okay, so that's that would lose a mark or two for that. Okay, next one here, onion cell magnification 00. I think the one got lost in the scanning. This is a fairly decent diagram. The shape of the cells hasn't quite planned that out. This cell here is a bit of a funny shape, so that's it's possible that you would find something like that. But the onion cells tend to have a little bit of that sort of six-sided shape. And so with this one sort of sticking into the other one here, it almost looks like they're overlapping, which they don't. Or it looks like this one's got sort of, an extra few sides to it. So what's happened here is they've obviously drawn this cell and then latched this one on. Okay, so plan out your entire diagram. Think about, that's the other main issue here, is that it goes off the page. So either the box the space they were given to draw this, or the page itself, they've reached the edge of that and just stopped. For a plan diagram, that's okay, because you're never going to be able to draw the entire structure. But for a cell diagram, we want basically 4 complete cells in the center of the page, not reaching the edge, but using the majority of the space that's provided. It also looks like this one's been done in pen, so that would lose another mark. Also in the nuclei, we've got a few lines that come around and stick off into the nuclei again. Okay, so being careful to connect those lines. Often when you're drawing small circles, that's when students do them really quickly. They do the outline of the cell very carefully, and in the small circles they just do them really fast, and you get these kind of this overlap or this line coming off inside again. Okay, so this diagram here, a few things. So firstly, some of the lines don't meet up. It's probably the scanning. Some of the lines sort of seem to fade out a little bit in a couple of areas. It's probably an artifact of the scanning, so I won't sort of focus on that. But here we've got a little bit of a double line. So need to be careful that those lines are really one on top of the other. And here there's possibly a slight gap. The other problem is this bottom cell really has just been latched on here. Okay, so it actually looks like the student's probably drawn this cell, then drawn this one on the side, and then added this one on the side here. So this looks like what we call like fish scales. It actually looks like they're overlapping, like one cell is sitting on top of the other cell. Especially since the cell here, its cell wall probably at this point would have probably followed this line and carried straight down, rather than sort of curve around the side like that. It's possible, but it's unlikely. And straight away, looking at this diagram, you can see it looks like this cell is on top, this is the next layer down, and this one is behind all of them. So you need to be careful that you don't get that kind of scaling or overlapping effect. So you need to really be careful, look at how you're doing your junctions, your corners, where the cells actually connect together. This one here is, again, I've put this in just as an example of the same thing. So the criticisms for this one, nice sharp lines, that's fairly good. The nuclei, we've got those lines, so it's always when students draw the smaller circles that they take less care than when they're drawing the whole cells, so those little lines sticking in. But actually mainly using this example, again, showing that scaling effect. All right, so the student's drawn 1, 2, 3 cells in a row here, and then added 2 on the side over here. So drawn in this one, and then drawn in this one here. So it actually looks like this one's behind these 3, and it looks like this one here at the bottom is behind all of them. It really probably is, if you look at it, this is a very unrealistic shape, that it kind of really comes in, and that it's got these 2 kind of massive protrusions here, like big horns. They do kind of spike often the cells, but to have these really 2 big, it really does look more like it's behind the other ones. So these cells look more like those fish scales, or those that they're layered rather than that they meet actually sort of side by side next to each other. So again, just something to be careful of there. Alright, this one here. Not a bad diagram, pretty decent. Little bit of smudging here. Pencil probably not quite sharp enough, getting a little bit feathery in a few areas. You can see some stuff here that was erased, but hasn't been erased completely. That may be the scanning might show that up a bit more than you could actually see on the page. The main issue with this diagram is that the cell wall has kind of just been left as a gap between the cells. So generally speaking, if you're looking at a cell layer, so anything that looks like a brick wall, like of plant cells, or even animal cells if they're stacked right next to each other. Animal cells tend to be too small to see like that, but so you'll tend to get plant cells. But if you've got cells that are in a cell layer, usually there aren't gaps, because usually if you think of like an epithelial layer, like a skin layer in animals, or any kind of cell layer, often it's supposed to act as a barrier, so a gap would kind of defeat the purpose of that layer. So the cell wall, if you're going to draw it, it needs to be drawn in. Each cell needs its own cell wall. You can't just put it in there as a gap. Alright, so this one here is just trying to show the same thing. Again, pencil here may not be quite sharp enough, getting a little bit feathery. You may need to sharpen your pencil during your drawing. Some of the other lines are, like here it's fine, or over here that's beautiful, that's nice and sharp, but then here it sort of starts to get a bit feathery. So, or especially down here, getting a bit feathery, sort of looks almost a bit smudged. So just being careful with that, maybe having to sharpen your pencil as you go. But the main thing here is that plant cells don't share a cell wall. So if you're going to draw in the cell wall as a double line, every cell needs its own one. So in the middle, they can't actually just be sharing it. It's not like a secretion. It's like its actual skin. So it's this one's kind of showing it. Think of it like skin, like it. Each cell has its own one. They're not sort of fused together, like it's sort of shown here. Also, you should be drawing the cell wall in proportion to the cell. So some plant cells have a very thick cell wall, and that's fine. You can draw it in nice and thick. But often it really just looks more like a line, and so it's probably easier that you do it just as single lines. Now this student has done some stippling in the nucleus, I guess just try and show that it's a bit darker. Like I've said in class, stippling is really generally to show texture, so it's not necessary, but at least the dots here are dots, they're not lines, so you wouldn't lose marks for that. Okay, this last one, good example of really just not using a sharp enough pencil. Should be drawing more cells. The cell shape is reasonable, although you can see here you've sort of got one line, and then this line has obviously been drawn it hasn't sort of curved round. You've actually got like a little intersection in here. It actually sticks out a little bit. So draw the line as one continuous line. Don't draw one side and then draw the other side like when you're drawing a square or a box. Follow the pencil all the way around. And like I said, you might need to sharpen your pencil. Here you can really see it's just not sharp enough. Also, this one the cell was a bit small, so remember to use that space. But plan it out. If you get too big, you'll hit the edges. You won't be able to fit in your 4 cells, and that can be a bit of a challenge too. Alright, getting towards the end here. This one's getting pretty good. So not a lot here to point out that's probably too badly. Would have preferred that they did the cell that was going to go here rather than this one. So remember that rule, trying to draw each cell touching 2 other cells. But it's okay, they'd probably get away with that. Here possibly the pencil wasn't sharp enough, although that could be the scanning. Just a little bit feathery there. Yeah, this one's actually a pretty decent drawing, and this probably wouldn't lose any, or would lose maybe 1 mark just for not sharp enough pencil. But like I said, that could be the scanning. I think this one's pretty decent. Needs to underline the heading though. Need to put that in there. But otherwise, yes, a very good diagram. This one here again is pretty decent. So again, it could be the scanning that's making this line here and here looking a little bit feathery. The shape of the cells isn't maybe great, looking a little bit sort of round here. They do often have a sort of a roughly sort of 6-sided. Even if all of them, like this one's kind of got it, and this one here sort of has it, although down the end here, possibly latching it on a little bit. But this one down here sort of loses that shape a little bit, becomes a bit round. So just be careful with that. Try to show if it's got a sort of a roughly 6-sided shape, but at the same time don't draw them as straight lines and connect them like when you're drawing a square or a triangle or something. You still need to be following that round as one continuous line. Okay, just a couple of tips when doing drawings. The first one that's really key is a really sharp pencil. You want that to be really sharp. Remember to bring a pencil sharpener. Okay, so when you're drawing lines, apply a decent amount and kind of consistent pressure. So draw the line. If you have to move your hand without taking the pen or the pencil off the page, you're less likely, you'll get less visible junctions than if you draw a line and then you try to connect up the other line. You kind of tend to get these little bits where it doesn't match up perfectly. The other bit of advice I can give you is if you have a line that, let's say, needs to connect onto this one here. Alright, so we're coming around. Don't try and meet it at a T kind of intersection. You'll either go over the line, and then you get what I've done here, a little bit sticking over the other side. Or if you don't go far enough, you'll actually have a tiny little gap, and you can be punished for that too. So what I recommend you do is, as you come or as you approach that line, to curve off and to follow the line. So you actually go along with the line for a short way. In real life, in biological sort of drawings or in real biological specimens, it's not usual that you would get a sort of a perfect T right angle junction anyway. So like I was saying, if I was coming down, draw one line, if I was coming down here and I needed to connect on, I would sort of curve slightly and join that line. Okay, and that just gives you a tidier join, or you don't get those crossing over lines, or you don't get a gap that students often lose marks for. Alright, the other bit of advice is really just, make sure your lines connect up. So for instance, if I'm doing a circle, we get a lot of circles that do kind of this. So we get this little crossover, or we get gaps. They don't match up perfectly. So you really just got to make sure, and again, it's about meeting the end and possibly just following it over a little bit, going really slowly where that join is. And so yes, you can still see possibly the junction there, but it is much more representative of what you actually see. Okay, so I hope those things helped. Also, just remember, if you make a mistake, you need to be really careful that you erase it out completely, so that there's really no trace of it left. Cool.